Bacteria found in wastewater possess the ability to decompose plastic as a source of nourishment. Greetings to everyone. Today's video will focus on the recent discovery made by researchers at Northwestern University regarding the Comomonas bacterium and its ability to decompose plastic for sustenance. Initially, this bacterium breaks down plastic into nanoplastics, after which it secretes an enzyme that facilitates further degradation, utilizing the carbon atoms as a nutrient source. This significant finding may pave the way for bacteria-based approaches to address plastic waste, particularly concerning polyethylene terephthalate, PET, which is a substantial contributor to environmental pollution. The study underscores the influence of wastewater in the formation of nanoplastics and highlights the necessity of comprehending the behavior of plastics within environmental processes. Without further delay, let us begin our discussion. Researchers from Northwestern University have recently unveiled the mechanism by which Comomonas bacterium cells decompose plastic for nourishment. Initially, these bacteria fragment the plastic into diminutive particles known as nanoplastics. Subsequently, they produce a specialized enzyme that further degrades the plastic. Ultimately, the findings reveal that the bacteria utilize a ring of carbon atoms derived from the plastic as a source of sustenance. This discovery presents new opportunities for the development of engineering solutions, utilizing bacteria to address the challenging issue of plastic waste removal, which contaminates drinking water and poses threats to wildlife. The research is scheduled for publication on Thursday, October 3rd in the journal Environmental Science and Technology. We have demonstrated systematically for the first time that a bacterium found in wastewater is capable of utilizing a plastic material as a carbon source by deteriorating, fragmenting, and breaking it down, stated Ludmila Aristilda from Northwestern, who spearheaded the research. It is remarkable that this bacterium can accomplish the entire process, and we have pinpointed a crucial enzyme that facilitates the degradation of plastic materials. This discovery could be optimized and harnessed to aid in the removal of plastics from the environment. Aristilda, an associate professor of environmental engineering at Northwestern's McCormick School of Engineering, specializes in the dynamics of organics within environmental processes. She is affiliated with several institutions, including the Center for Synthetic Biology, the International Institute for Nanotechnology, and the Paula M. Trenans Institute for Sustainability and Energy. The study's co-first authors are Rebecca Wilkes, who previously completed her PhD in Aristilda's lab, and Nanking Zhou, a current postdoctoral associate within the same laboratory. Additionally, numerous former graduate and undergraduate researchers from the Aristilda lab played a role in this research. The recent study expands upon earlier findings from Aristilda's research group, which elucidated the mechanisms that allow Comomonas testosterone to metabolize simple carbon compounds derived from the degradation of plants and plastics. In this latest investigation, Aristilda and her team focused once more on C-testosterone, which thrives on polyethylene terephthalate, PET, a widely utilized plastic in food packaging and beverage containers. Due to its resistance to degradation, PET significantly contributes to the issue of plastic pollution. It is essential to recognize that PET plastics constitute 12% of the overall global consumption of plastics, Aristilda stated. Furthermore, they are responsible for as much as 50% of microplastics found in wastewater. In order to gain a deeper insight into the interaction of C-testosterone with plastic and its feeding mechanisms, Aristilda and her team employed a combination of theoretical and experimental methodologies. Initially, they isolated the bacterium from wastewater and cultivated it on PET films and pellets. Subsequently, advanced microscopy techniques were utilized to monitor the alterations occurring on the surface of the plastic over time. Following this, the researchers analyzed the surrounding water for indications of plastic being decomposed into smaller, nano-sized fragments. Lastly, an investigation was conducted within the bacteria to identify the specific tools employed by the bacteria to facilitate the degradation of PET. Aristilda stated, "The microplastics were decomposed into minuscule nanoparticles of plastic in the presence of the bacterium. Our research revealed that the wastewater bacterium possesses a natural capacity to break down plastic to the level of monomers, which are small building blocks that combine to create polymers. These diminutive units serve as a bioavailable carbon source that bacteria can utilize for their growth. Following the confirmation that C. testosterone is capable of degrading plastics, Aristilda sought to understand the underlying mechanism. Utilizing omics techniques that assess all enzymes present within the cell, her team identified a particular enzyme that the bacterium produced in response to PET plastics. To investigate the function of this enzyme further, Aristilda requested that collaborators at Oak Ridge National Laboratory in Tennessee create bacterial cells lacking the ability to express it. Intriguingly, the bacteria exhibited a loss or significant reduction in their capacity to degrade plastic in the absence of this enzyme. While Aristilda envisions the potential application of this discovery for environmental remediation, she also notes that this newfound understanding can enhance comprehension of the evolution of plastics in wastewater. Wastewater serves as a significant reservoir for microplastics and nanoplastics, stated Aristilda. The common perception is that nanoplastics enter wastewater treatment facilities in their original form. However, our research indicates that nanoplastics can actually be generated during the wastewater treatment process due to microbial activity. This is an important consideration as society seeks to comprehend the behavior of plastics throughout their progression from wastewater to the rivers and lakes that receive them. The research, entitled Mechanisms of Polyethylene Terephthalate Pellet Fragmentation into Nanoplastics and Assimilable Carbons by Wastewater Comomonas, received funding from the National Science Foundation under award number G2109097. To support our channel's growth and ensure broader awareness, kindly hit the like, share and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.